All right, so here is chapter 27, part 3. So the last time we left off was when we were about to start talking about the nuclear envelope. So the leading hypothesis for the origination of the nuclear envelope is that it derived from infoldings of the plasma membrane. And along with the nuclear envelope, um, the infoldings also gave rise to the endoplasmic reticulum. And the evidence that support this is that infoldings in plasma membranes are also found in some living bacteria. So the nuclear envelope and endoplasmic reticulum of eukaryotes are continuous. So here you can see the ancestor of eukaryotes with this cell. The chromosomes are inside. You have the plasma membrane, and then kind of like when you press, like if you have a water balloon and you press your finger in it, it creates an infolding. So you have the infoldings forming on the outer edges, and they surround the chromosomes, and then eventually um, the infoldings form a nuclear envelope along with the the endoplasmic reticulum. So the reason this is important is because the nuclear envelope allowed for the separation of transcription and translation. So transcription occurs inside of the nucleus, and translation occurs outside of the nucleus. So this just gave early eukaryotes uh, a new way to process information. So with this, it allowed for the nuclei to diversify, leading to unique types of nuclei in different types of protist lineages, so different groups. The distinctive structure of the nucleus is the synapomorphy that distinguishes these lineages as a distinct monophyletic group. Structures for support and protection. So protists also have a lot of diversity in the types of uh, structures that they use for support or protection. So for example, the cell walls in things like diatoms are surrounded by glass-like um, structures that are made of silicon dioxide. With other protists like dinoflagellates, they have a cell wall that's made up of cellulose plates. In others, you can see um, hard external shells. You can see rigid structures inside the plasma membrane. And so the diversification of protists associated with evolution of innovative structures for support and protection. So here are some examples of the different types of the outer coverings of in protists. Okay, so here's the diatom with that silicon dioxide plates. These kind of fit together like a petri dish, where you have one plate um, that goes right on top of the other. Here's the dinoflagellate with the cellulose plates. Here's the foraminiferans with the calcium carbonate. So we talked about this one last time when we talked about some of the role that protists play in the carbon cycle. So when the protists die, or for these particular protists, when they die, they go to the bottom of the ocean and they form, um, essentially, they turn into rock, right? The protists along with other sediments that fall on top of them. So these are those foraminiferans that have the calcium carbonate. Okay? Okay, multicellularity. So mutations leading to multicellularity probably first cause cells to stick together after uh, dividing. Selection pressures acting on larger colonial organisms allowed them to evolve and diversify. And eventually, cells became specialized for different functions. Okay, so not all cells express the same genes. Multicellularity arose independently in a wide array of eukaryotic lineages. So how do protists obtain food? So similar to the way that we looked at, in a similar way that when we looked at the feeding habits of bacteria and archaea, so protists can also obtain food either by obtaining molecules from the environment or through things like photosynthesis. But the other thing that protists do that's different than bacteria is that they can also eat things with a process known as phagocytosis, where the individual takes packets of food much larger than individual molecules. So protists, so these are the three ways that protists feed. So they ingest large packets of food, the phagocytosis. Okay, so when one cell consumes another, they absorb organic molecules directly from the environment, and also they perform photosynthesis. Ingestive feeding. So ingestive uh, lifestyles are based on eating live or dead organisms or on scavenging loose bits of debris, of organic debris. Some protists are large enough to surround and ingest bacteria and archaea, and some are even large enough to ingest other protists or other small microscopic animals, so like small arthropods. Feeding by phagocytosis is possible in protists that lack a cell wall. So, for example, something like an amoeba. Okay? The flexible membrane and dynamic cytoskeleton allow them to surround and swallow the prey with their pseudopodia, which are just extensions of the cell. Okay? Many ingestive feeders are actively hunting for prey, and then there's others that kind of just sit there and wait for prey. Okay? And they obtain the prey by moving their cilia around their mouth to kind of create currents of water to sweep the food particles inside of their mouths. So like suspension feeders. Okay, so here's an example of the a protist that's actively hunting and consuming prey or trapping prey with those pseudopodia. So pseudo meaning false, podia meaning foot, because they're not true feet, but they look like feet. So they call it pseudopodia. And then here's an example of suspension feeding. So when a protist uses ciliary movement to sweep the food into their mouths. There's also absorptive feeding, where the nutrients are taken up across the plasma membrane directly from the environment. Some absorptive feeders are decomposers, so they feed on detritus. Detritus is just dead organic matter. And then there's many absorptive feeders that live inside other organisms. If you have an absorptive feeder living inside an organism and it's actually damaging the host, then you call it a parasite. So some examples of those parasitic protists would be like the ones that cause malaria. right? So they live inside mosquitoes and humans, but they're doing damage inside, so they're considered parasites. And then there's protists that do photosynthesis. So these are autotrophic protists, and these produce organic compounds via photosynthesis. There's also they also range in size, okay? So they can either be single-celled organisms like some algae to giant seaweeds, so like kelp. And these use carbon dioxide as a primary source of food. Okay, so very similar to plants, to land plants, that is. So how do protists move? Many protists actively move to find food or to find light. Okay, the first type of motion is called the amoeboid motion. So this is a sliding movement that's observed in some protists. It's very similar to the way that the muscle movements in animals. Um, and so, and it's also similar to the way that your immune system cells uh, move inside of your body. Okay, so here's an example of a protist using amoeboid motion. So they're using the pseudopodia to it kind of just extends forward and then it kind of drags, the rest of the body follows. The other major mode of locomotion is is achieved by using things like flagella or cilia. So these are types of organelles. So flagella and cilia, they're similar. The flagella are long and they're usually found alone or in pairs. So you'll see two flagella usually at the end of the animal or one. The cilia are usually short and they're usually surrounding the animal. Okay? Or the talking about protists here. Okay, so here's flagella. There's cilia. So in the cilia, this would be like the one that you saw in from lab when we looked at a paramecium. Okay, so paramecium, those are the ones that had cilia all the way around their body, or it was all the way around the cell. Okay, so how do protists reproduce? Let's see. Oh, okay, so yeah, you know what? We'll stop here and then we'll continue with the reproduction. So we'll save that for another uh, short video.